Hello friends, welcome to BMH Learning. Here we will talk about actin nucleation by Formin protein. So this is the structure of Formin protein. We have three domains here. First is the FH1 domain. This is proline rich region, and proline residues are binding sites for profiling bind to ATP actins. In other words, it mediates the recruitment of profiling ATP actin, right? Then we have FS2 domain. This domain nucleates assembly of actins into filaments. Apart from this, there is third domain too, which is named as RBD or row binding domain. Now, RBD is the main site which responds to extracellular signal. So let's discuss the mechanism. For now, the Formin protein is inactive because its FS2 domain is inaccessible. As the cell gets the signal for actin nucleation, GDP is exchanged for GTP, making Rho active. The now stimulated Rho calls for its binding partner, RBD domain. The binding between RBD domain and Rho exposes Formin's SS2 domain. Now, this structural rearrangement of Formin protein lays the foundation for actin nucleation. So, initially we have ADP actin, and with the binding of profiling, it becomes ATP actin by mechanisms discussed in detail in previous videos. Now the profiling ATP actin complex is ready. So one by one, they are recruited to FH1 domain. This way, FH1 behaves as a landing site to increase the local concentration of profiling ATP actin complexes. Then for the polymerization, with the concomitant release of profiling, the actins are fed into the FH2 domain to get polymerized at the plus end of actin filament, thereby allowing rapid FS2 mediated filament assembly. Now let's discuss the detail of actin nucleation process. So we have the first step, formation of FS2 dimer. That is, two FS2 domains from two individual monomers associate to form a donut-shaped complex. The dimer then binds two actin subunits in such a way that the plus end is towards the FS2 domain. Now concentrate. To further grow the filament, the FS2 domains move back and forth. So, to get hold of another actin monomer from FH1, first, one FS2 domain moves. This action lets go already imprisoned actin subunit, thus freeing up some space which allows the addition of next actin molecule. And then, second FS2 domain shows action, and we see the steps being repeated again and again, resulting in growth of the filament. This was all. Thanks for watching.